incubation temperature, autoclave, motility temperature, refrigeration. Refrigeration is 4 degrees Celsius.
So we need to see the little
vials,
vials,
cloudy and
the green one, and
Green.
that you they look like a bunch of mollusks or something other that all consume the whole thing.
wasn't that where the
Oh, the picture.
fragmented and am I completely off? Was there a
Have you
fragment?
the picture?
The thioglycolate tubes,
Yeah, you guys talking
or
about
the
the
sim
tubes?
tube, or the TTC tubes.
Was it the thiopsine?
I think it was the thiotubes.
You showed us the two where they had were anaerobic, aerobic, and how they could go. And then there was one, and it was either black or greenish, and it was completely consumed in the tube.
Yeah, that's motility. All right. Is everybody okay with the information in lab 2? Did everybody take pictures of all the slides and everything I put up so you could identify the stain and you know the difference between the simple stain, differential stain, examples, things like that. In lab 3, we talked about a lot of different ways for motility to be observed. Everybody remember that swarming plague? The swarming plague was where there was a dab of organism put in the center and it moved around and made kind of circles. You guys remember that one?
Yeah.
Now the sim tube also identifies motility. Now, it's if it moves out from the stab line and it's cloudy out from the stab line. I tend not to use that one as much as I use this sim tube because it's a sulfur reducer or H2S producer. So if you always on the top, no matter what tape I cover it up with, it'll say sim media.
Let's say what.
But you know the M is for motile. So that black means that it is motile. It's also an H2S producer, which is not as big of a concern until lab 7, but motility is what you need to associate with this particular type of organism. Okay. Now the TTC tubes had a little bit of PKs outside of the stab line. You guys remember those? It's a little harder to detect. E. coli was motile, but was not. I'm not going to ask you the names of them. You guys remember this one?
Am I taking pictures of those?
Did anybody take pictures of these?
I got a few.
I didn't because I didn't have
I went
one.
through the pictures I had, and I sure didn't have near as many as I thought I
I'll
had.
send you what I got. I'll send all of you what I got.
You got those? Okay. Now also flagellar arrangement. I think everybody was okay with flagellar arrangement. You guys have been tested on that already in class. And then remember I had the spiral organism that was Amphitrachus that had a flagellum lesion in that lab 3 I showed you. Lab 4 was more so the aerobic, anaerobic, facultative, all these atmospheric oxygen needs. This particular one, Clostridium, was clear at the top, cloudy at the bottom. Did anybody take pictures of these?
I'm coming that way. You had those, Terry?
Kind of hard because you can't lay it down, so I mean, I can hold it up if you want to try to get the right angle on it, I don't know. But this is an anaerobe. Now the E. coli, that was an antler. E. coli was a little rough because it only had a couple little speckles at the top. The bacillus was a little more so cloudier throughout. You can see a little more. some sides you can see it goes all the way up. So that one's a little rough. You know. That one was facultative, and I didn't have an arrow. And I won't have an arrow ever for a month of class. Because it'll die over the weekend and it'll start trickling down, and then it looks like it's. Right, right. And that one still, it goes up further than the E. coli does, but it's still little kind of dots almost at the top. Lab 5 was where you probably learned the majority of the stuff as far as media goes, like the MSA plate, blood auger. I didn't do any additional blood augers. Didn't have the time, to be quite honest, so I might ask you a theoretical question like what color is it if it's alpha? What is it? Yeah, this is the diagrams with the MSA plate, the McConkey plate, the blood auger.
On there. Take picture of the mouth. You're laughing at we were gagging.
Yeah, I know.
Yeah. Yeah. You got, that's the MAC, the fermenter versus non.
I was gonna ask you what these were, because I took a picture, but I don't have any reference to, so what are they? That's terrible.
That's the MAC plate, fermenter, and non-fermenter. And see, if you look, you can kind of see where it says McConkey 2.
Okay.
McConkey 2, that one's upside down. So the fermenter, remember, was.
I don't have yellow. I'll make a copy of mine.
If you don't mind. I think the only blood auger I showed you was the only real good one that I had was the, maybe I'll, nah, I'm not gonna have Brandy make any more for me because you guys won't be able to see it. So just,
And that was,
that is the blood auger, and that's showing beta hemolysis, how it's clear around where the, you see the first streak, second, and the third, that clear border is showing you that it's beta hemolytic. That's the only one I'll put on the test. It's the only one that I showed everybody, so in all fairness, that's just what we
No, I
got.
found mine.
blood auger, beta hemolytic, right?
Mm-hmm.
Okay.
Mind if I can make copies real quick?
Go ahead. And then you should have an MSA plate that's real yellow and then one's pink. Phone's nice and thin.
That's the first one we just did. This was the fermenter, non-fermenter, right?
That's the McConkey. You should have an MSA plate though. Fermenter's yellow in that one. No,
That's the same one, isn't it?
that's the McConkey. The MSA.
I don't think I have it. I have to get it from one of them. This is the same as that other plate,
Yeah.
right? Just a different.
Yeah, I'm not gonna show you that one. That one was throat.
Okay, we'll forget that then. And then I'll just have
Yeah,
these.
that's all you got.
I know it took more. I just don't know what I did with them.
I guess I left them out in the car.
Is this last slide I just showed you, was that gonna be?
Yeah, that's the Amphitrachus organism showing flash a little ring. So, yeah, very possible that one.
Okay.
What for the MSA?
His name.
That's the fermenter, not.
Said multiple. This has multiple.
One on each end, yeah. Yeah, it's that one that goes around like that. Yeah, I'm not sure what that other artifact is, but yeah, that one's good, that sure can change it, kind of fold it over. And that should be all in place once you got that one. So I don't know how you guys do it, if you post on Facebook or anything like that. Because people do that, do the pictures,
Email and YouTube. So you see Dr. Mike do a little bit on YouTube.
the MSA. Fermenter, not fermenter. It's just that or is that heavy. Well, that's a good thing we talked about that. There was
Yeah.
a couple things you guys didn't quite have. Okay.
The
Wow.
fermenter was pink, right? On that MSA plate, the fermenter was pink, the non-fermenter was the yellowish color.
The fermenter is yellow.
Oops, I had it backwards. Just the opposite of the other one then, right?
Yep.
Ooh, glad I asked.
Because that plate starts out pink. So that change in color means it's a fermenter.
Let me write that down. That will help me to remember.
Let's
Plates. That's why I said plate.
So make sure in that lab 5 you look to see the different media types and how they're classified. So if I ask you an example of a minimal basal auger, complex and differential, selective and differential, you know what's what. So that's a very, very, very important chapter. And also the diagrams that you drew were very important because I'll ask you quite a few questions on
Yeah,
those.
just a star that
Yeah.
highlight.
So make sure you got those down. Again, you can write them on the back. I'm okay with that. So, however you would prefer to do it.
My brain is in slow motion on that. Diagram from chapter 5.
Yeah,
Lab 5.
these ones.
Yeah, lab 5.
Mm-hmm.
These ones.
Okay.
All right. Any other questions about the lab? I think we kind of tidied up a little bit of those things that you didn't have.
Did you ask her about cleaning the off?
Mhm.
They said on the lab sheet,
Uh-huh.
it said that you clean your desk off before and after. Are you
Yeah,
talking
and
about like
With the back
after?
down, right. I never make you guys do it before, but after because it should be always clean. But
So for the test,
for the test, yeah, technically both, yeah. So how it's worded in there is, we do not have any other labs besides ours and any of my other micro labs that wouldn't be doing that cleaning. So it is a good idea. Some people still clean before. I mean, that's always nice to do. But we make sure we always clean up before we leave. And the AMP people don't do anything but so it's kind of a mixed lab, and that's why I kind of bend on that a little bit. But of course, I don't know what Mr. McNichol's lab does. We should probably go ahead and clean before and after. You never can tell sometimes. Yeah, well. All right, any other lab questions? So the rules, yeah, definitely look to see how they're worded in the actual lab, and I just take stuff from the lab. And if not, we'll get on to chapter 9. You guys ready?
oh yeah.
Any other questions?
Actually, it'll take about 3 minutes.
Yeah. Take about 3 minutes.
Takes you 3 minutes? You got PPH?
I gotta wash my hands real well. Gotta sing the ABC song.
Gee,
It takes
3
nine.
minutes? And you know, I got a pile in here, guys, if you're all stuffed with big stuff. Here again.
We'll let you slide.
Yeah.
Tried to keep up with everybody that gave me a quiz through email. I entered 2 years today. Yeah, I think it was chapter 8 and chapter 9, right?
Yeah.
I was like, dang, she's already
I
posted
did
9.
one.
And
then gave
you gave me a hard
same
copy
thing.
of it, yeah.
Yeah, some tripping.
So I got that. That's
Paranoid.
sometimes with I mean, I prefer them in hard copy because sometimes, man, you gotta remind me to get those in with the email. But I try to do them when I see them, so that I don't miss anything along the way. But hopefully, if you don't see something, just let me know. And I'll look in my email. I don't delete them, so I just keep them there till I do it.
Yeah, I was shocked. I was like, dang, she already posted it. Yes, so back to number 2.
Oh, that's so funny.
Did you by chance grade my test?
I haven't graded yours and Taria's yet. No, I had tons of other makeups that I had to end up doing. And it was a little bit of a race to get the final grades in. People are still trying to do makeups even though I've turned in final grades. And it's been a week since the exam.
Wow.
It's just, I had to finally turn in grades, so I don't know what they're going to do. I'm over it.
See you next term
Right.
or next quarter.
Well, I don't know why you'd wait a week till the exam. And probably somebody heard that somebody got, because there were some people that did have some valid reasons, but I couldn't take one over the other because it's a final exam. I don't do any makeups for those, so it had to be some type of excused type of absence that the department heads, either over nursing or over general studies, they have to approve it, send it to me, and then there you go. So you get all these people that hear through the grapevine that somebody else did it. They didn't necessarily possibly have those requirements that so I don't know how it's just constant. I can't ever finish anything up.
That is so annoying.
Yeah, I mean, when you take a makeup exam, you're only supposed to be allowed to take one, and a lot of these people have taken more than one, so that's why they maybe did not qualify, and nobody sent me an email to. They may not understand that they can't. I don't know. I got no clue. People don't listen to rules very well, so it just doesn't. So it's not that I've just been in a constant
Well, I knew with the exams last week that it was.
constant chaos. Yeah, supposed to have. We typically turn it in like exam week Friday at 5 is typically when we turn it in. Sometimes they'll extend it Monday to 5. So I kind of tried to stay within those guidelines, but who knows what they'll do. So these, I have, which one do you want?
That one.
This one right here?
No, yeah,
The other one?
yeah, that one's better.
There you go.
That's what I thought. So, in all probability, because then I gotta I gotta figure out what I got for the next term. At least Wednesday, on Wednesday I will get this class balanced out again. Not that you guys are last on my list, because you know I like you much better than the
Yeah.
quarter people, but.
It's not like I'm stressing over it. I know
No one's free.
so, you know.
Alright. Well, let's get chapter 9. You guys know chapter 10 is on the test. We went through this before, central dogma of biology. Keep in mind that, of course, with DNA, You want to pass that information on to a new cell, whether it be through mitosis or so forth. You got to have an exact copy. Once you got that DNA in your cell, after it's went through mitosis, you'll have a need to make proteins. But in order to make proteins, you have to go through the process of transcription, which, remember, uses one side of DNA. And it makes messenger RNA, which is only single-stranded. And then with that messenger RNA and translation, actually all three RNAs play a role and put together the final product, which is the protein. So transcription and translation is part of protein synthesis, where that DNA replication has to be done only when you're passing that genetic information on to a new cell. And of course that DNA is vital for protein synthesis because it has the information. Any questions about this? This is old news. Genome, that's just the total DNA of a cell. Especially when we talk about prokaryotic organisms, a lot of those genomes end up being circular. Some eukaryotic microorganisms may have a linear chromosome. So I like to use a linear model just because it's easier to take a look at as far as replication goes. Plasmids, keep in mind that some organisms may have plasmids. Those are circular and they're extra chromosomal DNA. So they might encode for some sort of antibiotic resistance factor or something. It's a little bit like evolution in a way. It's got some type of encoding that helps make it resistant to some type of environmental condition. So a little more easier for it to survive. Antibiotic resistance is one of the obvious examples of that. And remember, plasmids can be transferred through that sex pilus that we talked about in chapter 4. So you might have an organism that has its chromosome, then they have some extra chromosomal information that's going to help it survive different conditions that other organisms don't have. Genotype, that's the genetic makeup. The phenotype is going to be the expression of it, so it would be the appearance and function. I usually don't ask for genotype and phenotype. We reviewed the other day, so hopefully we can get through this really quick, the structure of DNA. You guys remember the double-stranded versus the single-stranded RNA. What were the differences between DNA and RNA?
Deoxy.
The deoxyribose is the sugar in DNA, ribose is the sugar in RNA. What else?
Was it A and U?
The T is in DNA. So thymine we find in DNA, but uracil we find in RNA. You guys remember that? And then it's just the double-stranded versus the single-stranded. Now, remember that DNA is anti-parallel. So when I use these terms, one side runs 3 to 5, the other runs 5 to 3. That's how that backbone's connecting together. I'm not going to draw you a picture. There's no sense in doing that. But one side runs 3 to 5, the other runs 5 to 3, and that means that it is anti-parallel. And all the 3 and the 5 are, remember that deoxyribose has 5 carbons. So it's just the 3 position and the 5 position where those bonds are connecting sugar, phosphate, sugar, phosphate, sugar, phosphate. Does everybody kind of remember that anti-parallelism there? And I have that on this side, and remember, it looks flipped over if you look at the other side of DNA. So they just run opposite, but they still connect 3 and 5, 3 and 5, 3 and 5, or 5 and 3, but it's still the 3 position and 5 position. Replication, we already talked about this. You need an accurate copy to be passed on. As far as protein synthesis goes, you got to have your DNA, and then that information is sent into the form of messenger RNA or transcribed into and then it's translated into the protein. That's just kind of a redo of what we talked about before. If we take a look here at DNA replication, there's only a small portion of DNA that's replicated at a time. So you work on a little bit, and then you work on the next chunk, and then you work on the next chunk. If it all separated at once, it'd be a big mess. So just done a little bit at a time. Now, as far as this term semi-conservative replication, remember I kind of hit on this the other day. Semi-conservative replication through several experiments, this is how they figured that DNA replicated, is that you end up having an old strand, which serves as your template. And then you have a new strand that's built alongside of it, and that's your complementary strand. Now that's different from conservative, where you have two old and two new, or dispersive, where you have little chunks of each spread throughout. So semi-conservative replication simply means you end up with an old and a new. So you could tell right here, once those separate, those colors are indicating this is indicating the new one versus the old one. So when those separate completely, you'll have a new and an old. Now, as far as the replication fork, that is the busy section. So that's where we're matching up all of these different nucleotides. That's where all of our work is taking place. Once that replication fork is completed, a new replication fork is created, and it moves on down the line until it separates the whole thing. So the first enzyme that I want you to know in this fork is called DNA helicase. And that's going to be the unwinding enzyme. And that exposes those nucleotides so that complementary nucleotides will match up. Now, DNA polymerase III, we're going to talk generically about this. And I'm going to show you a little something that happens that your book doesn't go through in quite as bit of detail as it should. But DNA polymerase III lengthens the strand in one direction, so it goes in the 5 to 3 direction, which means that its template would be running what way? 3 to 5, that's right. And then, of course, once that side's built, that DNA helicase will keep moving that fork. Now, this is the leading strand that uses that DNA polymerase III straight through. The other side will use DNA polymerase III, but it's built in chunks. So let's look here. You don't have to, we've already talked, you don't have to know about pyrophosphate being released, but you know what complementary nucleotides would match. If there was an A, it'd be a T. If there was a G, it'd be a C, and so forth. Now, taking a look at this particular, I like linear models because it kind of shows you what's happening. There's a leading strand and a lagging strand. The leading strand is the one at the top, and you see how that DNA polymerase III just goes really smoothly. And the arrow is pointing towards that replication fork. That orange ball is the DNA helicase, so as soon as that's finished, what's that helicase going to do?
When it's still in the polymerase III.
Yeah, it's going to unwind more, and then you're going to keep going. So see how this DNA polymerase III is moving towards that fork. You guys see that? Now, in the lagging strand, you got a couple of extra enzymes that have to come in and help. You have a primase, and the primase lays down these RNA primers. Now, actually, in the leading strand, there is a primase that comes in right at the beginning, lays down that primer.
I guess.
Oh, is it locked?
You said primary what?
In the in the leading strand, there is a primase that actually comes in at the beginning, just to get it started. And then DNA polymerase 1 destroys that RNA and cleans it out. But the lagging strand is the one that has the problems. So, in the lagging strand, the primase is going to make RNA primers in chunks all across that lagging strand. You got DNA polymerase 1 that helps to clean up those primers, but what happens is DNA polymerase 3 can run for just a little bit, then it stops. Then another primer adds, or another primase adds a primer, which is RNA. DNA polymerase 3 runs for a little bit and it stops. And then DNA polymerase 1 comes in and cleans up all those primers and lays down DNA. So the lagging strand is done in chunks and it takes a lot more time. There is a DNA ligase that's required for this particular lagging strand, and that's going to seal up the fragments. Now DNA polymerase 3, because it can only go in one direction, goes the opposite way. It goes against the replication force. That's scary. So to try to help you out, these keep in mind here with your leading strand, this DNA polymerase 3 plays the largest role. So once you have a primer right at the beginning, then DNA polymerase 3 can go forever. So it's built really, really quickly. This is something that a lot of the other textbooks don't teach you, but I'm not going to ask you about the primer at the beginning of this portion right here. But keep in mind, I'm just telling you guys that so you know. With the leading strand, it does have to have a little start, and that primer starts it. And then DNA polymerase 1 comes in and cleans it up. But DNA polymerase 3 just goes smooth. This little guy here, the lagging strand, there is primase comes in, makes all these RNA primers in chunks, so it happens all the way through. DNA polymerase 3 runs the opposite direction against the fork. DNA polymerase 1 has to come behind and clean up these primers. The primer, kind of think of it like a primer for paint, gets it started. So without that primer, DNA polymerase 3 doesn't know to go. Does everybody kind of get me there? So even though primase and DNA polymerase 1 doesn't play a very large role up there in the leading strand, but in this strand, primer, DNA polymerase 3, and DNA polymerase 1 throughout the whole strand. Kind of get that? So in this way, you would say have a primer. DNA polymerase 3 would go for a while. You'd have a primer, DNA polymerase 3 would go for a while. Whereas this side going in, DNA polymerase 3 go the whole time.
So if it's continually unwinding though, with the primer, I mean, because it stops for a little bit, I mean, is it getting regenerated up at this end and coming down? I mean, how's it, I mean.
This leading strand doesn't need a primer again.
Right, the lagging strand now.
Yeah, it needs it the whole way through.
Okay, so it's kind of.
The whole way through. And then polymerase 1 comes in and cleans these old primers up. So it's a lot of extra work here. And that's why it's the lagging strand. It's going to take a little longer because you got more enzymes that come in and have to do a little more work. And the ligase seals everything up.
And the polymerase 1 cleans up the primers, right?
Right. So up here, you got a primer that just comes in initially, and that DNA polymerase cleans it up, but that's all you got. So you got 1, compared to multiple sides all the way down.
So it's like doing the door and the hood and whatever.
That's right. That's right. So you gotta stop after DNA polymerase 3 will only go for a certain section, then it doesn't recognize it, you gotta have another primer, and then it goes again. So it is very, very complex. I might have a picture of that. Kind of sucks that we are not. Can't even remember some of the names. Where are they off on this one? We're not using this book again anyway, so. That's pictures of conservative and semi-conservative and dispersive replication. Just to make sure that you guys know what that semi-conservative is, because they usually ask that. And this right here is a picture that shows you the leading strand and how you have to have a primer just to get it going. And then this will show you how these are actually made in chunks in the lagging strand. And you see your DNA polymerase 1 just took out a primer there. And that's what it's doing, that's its job. A little complex. And in your alls, in that one table that I wanted you guys to know. Yeah, table 9.1, removing primers, closing gap, or repairing mismatches. So gives you a really good definition of that DNA polymerase 1. Page 238. That's okay there. Any questions? So remember, primer, the polymerase 1, the ligase, throughout that whole lagging strand, making it a lot longer of a process. DNA polymerase 3, though, still plays a role in that, but it only goes for a little bit, then it'll go again for a little. DNA polymerase 3 moves towards the replication fork in the leading strand. It moves away from the replication fork in the lagging strand. Okay, any questions about replication? Now, as far as these circular chromosomes go, you need to know that there's a origin of replication. So in the circular, it's going to be the same type of scenario as the other linear form. But you got a bubble that forms. Replication will go in both directions. It continues until those forks meet. And there's a terminus, which means you're done, and they separate. So that's the only weird thing about applying it, is that you're going in both directions around the circle. Applying that linear to a circular is a little different. Of course, you're still going this way in a linear, so it's still kind of the same thing. Right, now in transcription, you guys need to know the 3 types of RNA: messenger, transfer, and ribosomal RNA. Now remember, in the RNA, no thymine, only uracil. Now the enzyme that we had in replication was a DNA polymerase, and there were multiple DNA polymerases. And think of it, remember I told you what polymerase was: polymerize in just easy terms.
Building.
Just building, right? So DNA polymerases build DNA. RNA polymerase will build RNA. So it'll polymerize it. Now RNA polymerase, I want you to know that as the enzyme. This is a lot more simplistic. In the process of transcription, it binds to DNA to the start site, which is called the promoter. It's supposed to be OR. Now, in DNA, you're going to have tons of different start sites depending upon which protein that you are trying to make. Each gene will encode for a protein. So you'll have promoters all along, depending upon what you are actually transcribing at that point. So promoter is the start site, RNA polymerase is the enzyme. Now as DNA unwinds, those bases are exposed. The complementary nucleotides will match up, but they'll be ribonucleotides in this scenario. So you'll have a different sugar, ribose, and you'll have a uracil instead of the thymine. Now you don't have to know the direction of transcription, 5 to 3. That's typical direction. You can see that only one side is being used. So you're only making one chain. Now, just like there is a start, there is a termination sequence that says you're done, because you don't want to run into the other portions. So that termination sequence indicates that transcription is over, that transcript is then released, that DNA will close up. Now, remember that if you are a eukaryotic cell, this still occurs in the nucleus like replication did, because your DNA is in the nucleus. That messenger RNA travels through a nuclear pore to get outside to the cytoplasm. Now remember, according to the chart that I put up on the board the other day, that everything for that prokaryotic cell is happening in the cytoplasm as far as replication and transcription and translation go. We okay with transcription? Transcription is nice and easy. Now, the three types of RNA, that messenger RNA, as far as the description, it's a template that encodes a protein. The transfer RNA transfers amino acids used to build the protein. The ribosomal RNA is actually part of the ribosome that supports protein synthesis. So ribosomal RNA is in the ribosome. So all three of those RNAs play a role in the next step called translation. Now when they draw these out, transfer RNA always looks like a T. There's an amino acid at one end, and this is an anticodon at the other end. Messenger RNA has codons all the way through it, and that ribosomal RNA is part of the ribosome itself. It looks like keys or something, like a big attached keychain. All right, now going to translation. Any questions on transcription? We okay there? Translation. This is where you're actually making the proteins. So you're putting amino acid together with another amino acid. Now as far as the genetic code, during translation you always have a sequence of 3 nucleotides. The 3 nucleotides that appear on that messenger RNA, that's called the codon. Now the anticodon is the complementary 3 nucleotides on the transfer RNA. So the codon is on the messenger RNA. The anticodon is on the transfer RNA. Now this chart is on page 246 if you take a look at it. Now since there are 3 nucleotides, this particular side gives you the first nucleotide, the second base, and the third base over here. Now your amino acids you'll find correspond to certain codons. You do not have to memorize this chart, bless you, or do you have to use this chart? I just want you guys to understand how this works. Now, let's take the start codon, for instance, because I want you to know the start codon. The start codon is AUG. So you look at the first base position, and I say, okay, there's my A. The second base, okay, there's my U. I come down, and then over here is a G. I know it's hard to see, but that strip right there. So AUG, that's my start codon. That encodes for the amino acid methionine. Does everybody see how that kind of works? Everybody okay with that? I think we did this in AP, didn't we?
I
How to
remember
use that
this.
chart. Yeah, so your first base right here, so that's an A. You just hold your position. We want a U, come down, and we want a G.
Okay.
So there's your amino acid. Now there's also several stop codons. Does everybody see where it says stop on that chart? Those are nonsense codons. So that term that I have up there, I want you to know that a nonsense codon doesn't correspond to an anticodon, so that means stop, you're done. So there's always a start and a stop. That AUG is typically the start codon for probably 90-some% of those proteins that are built. Not 100%, but majority of the time. Now, taking a look, and the concentration is really on the messenger RNA and on the transfer RNA, because remember that ribosomal RNA is part of the ribosome, so you can't really see it. But let's say the messenger RNA looks like a worm, and the transfer RNA looks like a T. Now this start codon AUG, we know that's for methionine. The anticodon would be what? UAC, so it's complementary. So A always goes with U, G always goes with C. So throughout the test, and once you kind of get that, that's easy. I'll ask you, I'll give you a codon, you got to give me the anticodon. So forth. Now, you take a look at where the anticodon's located, so that's going to match up with the codon, meaning that's the correct amino acid for that position. At the top of the transfer RNA is where the amino acid actually is. So the start is methionine, so I expect that amino acid methionine to be at the top. Now ATP is used for this process, just so you guys know. Now this comes, I think, from the first edition. You guys have some of these pictures all on one page, I think 248. Still goes in all these steps. I just want you guys to see how this works. The large subunit, you guys can see the large subunit outline, the small subunit of the ribosome. Those can disengage, so they come together when you're actually undergoing protein synthesis. Messenger RNA will bind to a portion of that ribosome. You don't have to know Shine-Dalgarno sequence, but it binds to a portion of that ribosome. And there are 3 different sites in the ribosome, one called the E site, one called the P site, one called the A site. Just think of the EPA. Now the E site is the exit site. And as soon as those transfer RNAs drop off their amino acid, they leave through the exit site, they can actually be reused again if that amino acid comes up. Now, if you take a look at these first steps here, what happens is you'll have this first transfer RNA, and see how it's attached to the codon. Remember, codon, anticodon. Does everybody see that? That first one moves over to the P position. The second transfer RNA comes in, brings its amino acid. Now, what do you think has to happen between those 2 amino acids at the top?
Break the bond. You have to break the bond?
Well, a bond has to form.
Okay.
Remember, proteins are just a bunch of amino acids strung together with a peptide bond in between each amino acid. So if I want to build a protein, I got to have 2 right there together so that a bond can be placed between. So if you look at the next picture, and this is going along with what you guys have in your book, you look at the next picture, I see that a peptide bond is formed between the first amino acid and the second amino acid. What happens is this last amino acid that came in, all of these amino acids, or last transfer RNA, sorry, all of the amino acids will be transferred to it. This little guy has already dropped off its amino acid, so it'll leave through the exit site. This guy in the number 2 in the A position move over to the P. The third one will come into the A. And then what happens between the second and the third? Peptide bond forms, right? It's all transferred to that last one. This little guy will leave through the exit. This one will move to the P. The new one will come into the A, so it's just an assembly line. Does everybody see how that, and it grows 1 amino acid at a time. So you got to know they're joined by a peptide bond. It moves 1 codon, which brings in 1 amino acid. The transfer RNAs are discharged at the E site and can be reused. So I was trying to explain up there at the top, and the pictures are, these are probably the best pictures that I've actually had to explain this process. Pictures are very self-explanatory. Now look at the, you guys see how the messenger RNA is kind of moving out? Kind of like on a sewing machine. Once you finish part of it,
you got a little bit hanging off the end, and it just keeps going on down the line. Now this whole process will continue until you get to a stop codon or a nonsense codon. Transfer RNAs don't match up. Ribosomes removed, protein chain is released, can fold, combine with another chain or so forth, and that process is complete. This is an old picture kind of showing you a little bit of the same thing. They don't draw it as well as your textbook does, so I'm a little preferential to that, page 248. I even use that with my new textbook because I really like it. Is everybody okay with that figure and can kind of get an idea of what's going on? I mean, the whole point is, you know that there's 3 sites. You know there's an exit site. You know that that messenger RNA is moving one codon at a time. That transfer RNA is bringing in that complementary anticodon with an amino acid at the top. Once you got 2 amino acids next to each other, peptide bond has to form. Transfer to that last amino acid, it moves over, the other one leaves, another one comes in. And the process keeps going until that chain is complete. Any questions? We good? Okay. Now, as far as transcription and translation being simultaneous, the only cells that can occur in is a prokaryotic cell. So I want you to know bacteria, for instance, can have transcription and translation simultaneously. You can have that chain transcribed and then translation will start. So it can start while it's going. And that's because there's no nuclear membrane. With a eukaryotic cell, remember, you got to finish transcription and then send it out through a nuclear pore into the cytoplasm. With a bacterial cell, there's no barrier there. So it can start as soon as that transcription starts. So those proteins theoretically could be built rather quickly compared to a eukaryotic cell, as it can go on together. The lac operon, I'm not going to go through too much of this here. This is a little too much for what I'm going to test you on. I do want you to know what an operon is, and it's genes that control some type of genetic function. They are inducible or repressible, which means they are allosterically regulated. Now, the lac operon controls lactose metabolism for E. coli. And again, Chapter 8, Chapter 9, they use E. coli as the example because that's probably the most studied bacterial cell. Now, typically, don't worry about the regulator, control locus, structural locus. Go down to the bottom where it says it's normally off. So it's typically off. And E. coli will use everything else that it can before it gets to lactose. Once it's starved of every carbon source, then that can be turned on and lactose may be used. But it still has that allosteric regulation that we talked about in Chapter 8. Does everybody remember?
I was going to ask you, I don't remember allosteric regulation. Sorry.
Well, remember you got an enzyme, just generically. There's 2 sites, a catalytic site. The catalytic site binds the substrate. The substrate is the substance that the actual enzyme is acting upon, so if it's changing into something else. The allosteric site, sometimes called the effector site, is going to be a molecule that binds to either let that activity of the enzyme go or to stop the activity of the enzyme. So that's what I mean by it can be induced or repressed. Now the effector binds to that allosteric site or effector site. That's why some people call it the effector site, because you have an effector that binds and says stop, go, stop, go. So the top site, that's what the enzyme's going to act on. So it'll change it to something else. This says you can go ahead and do that, or you can't. Right, as far as regulation of genes, I don't think I'm going to ask you this on the test, but you can actually regulate during transcription. You can regulate at the initiation portion, so at the start. If you have regulatory proteins that'll bind near that start site called the promoter site, that can change whether it's going to happen or not. Attenuation, you hear a lot of times that I say the strains are attenuated. A lot of times that involves something called an attenuation loop where it has to go uphill and down. So most of the time that protein isn't made completely because that RNA isn't transcribed in full. Translation, you can stop at initiation, elongation, or termination. So that's at the start while that chain's growing, or at the end. I'm not going to ask you that stuff. So what I'm saying is you can control at several different spots there on each of those. As far as mutations go, now this is fairly easy. You can have chemical mutagens, physical mutagens, biological mutagens. The one that I tend to point out is UV lines. There's different types of mutations. Sometimes they vary with what they're called in different textbooks. Sometimes you can have the wrong nucleotide put in. Sometimes there's different sequences that are deleted or a reverse order is put in. Transposition is where a segment of DNA moves to another area. Sometimes you accidentally duplicate a segment. So there's all different kinds of things that can actually go wrong as far as that goes. As far as the results of a mutation, they can only be conditionally expressed, so maybe sometimes it's not optimal for the cell, or it can be something that encodes for a protein that's actually lethal for that cell, so it might die as a result of that mutation. So they can vary as far as what they actually do. And the introns and exons that we did not talk about in your book, but there's certain parts that you're actually coding for that's a little more important than a non-coding region. So they can really vary as far as what part is affected. Don't worry. Now, I do want you to know what happens during UV damage. So with, let's say, exposure to UV light, you have two thymines that are together. They end up forming a dimer. So that means those thymines connect together on the same side. You do have repair systems that will be activated. But look at what the thymine dimer would do, the top picture, to that actual DNA. It'll distort it because they want to connect together on one side as opposed to the opposite side. So your repair enzymes come in, no matter what organism it is, remove that particular DNA that is affected. And let's say an incorrect nucleotide is put into the sequence. That's where you have a mutation that would result. Once that repair enzyme has come in, you're pretty much stuck with what you got. I don't really talk about transposition here, but the top three ways that DNA can be transferred in or out of a bacterial cell. Transformation, this is where DNA would exit a cell and is taken up by another. In nature or a natural environment there, very few bacteria have the ability to take up DNA fragments, so the odds of that happening would be very slim to none. As far as artificially, that would happen more so in the laboratory, as it would actually be induced for that organism to actually take up some DNA. Conjugated plasmids, remember I said at the beginning, plasmids are extra chromosomal DNA. They encode for some type of beneficial factor for the organism so that it can survive certain conditions. So that plasmid transfer through the sex pelus that we talked about in chapter 4, that would actually, first that plasmid's replicated, then through the sex pelus it's sent to the new organism. They sometimes call the organism that has that plasmid F positive, the one that lacks the plasmid F negative. Once it's replicated and sent through, both of those cells end up being F positive, so both have that conjugated plasmid. Transduction, using a bacteriophage, a virus that infects bacteria. It can reproduce in the bacteria. Sometimes those phages will get some of that bacterial DNA. That cell lyses, releases any of the normal phages or any of the ones that will have that extra bacterial DNA in it. That phage will go and infect another bacterial cell, inject that other bacteria DNA into its chromosome, so now it has DNA from another cell by way of a virus, so via a virus. And that's all. All right, questions? Well, this one's better, I think, than chapter 8. Chapter 8,
Yeah.
Chapter 8 and chapter 9 are both kind of difficult material, but that one's a little easier than the other one. Right. And we are finished with this particular chapter. I guess, where are you guys at?